Scientists believe that influenza virus is spread from person to person through exposure to large respiratory droplets, direct contact, or airborne dispersal. Infection takes place mainly in the respiratory tract. Infection begins with attachment of virus proteins to a receptor on the surface of the host cell. The virus is then taken in the cell by receptor-mediated endocytosis. As the virus enters the cell, the virus is internalized in a membrane-bound capture vesicle that carries the viral core. The vesicle is transported on microtubules inside the cell by host proteins called kinesins. During transport, the membrane of the vesicle fuses with the membrane of the virus, and the capsid undergoes uncoating. The viral core, RNA, and proteins are then released into the cytoplasm, where they are guided by host proteins to the nucleus of the host cell. At the nuclear membrane, the viral core uses host protein channels to enter. Inside the nucleus, cell machinery is utilized by the virus to replicate the viral genome and make messenger RNA, mRNA. Some viral mRNA exits the nucleus to exploit cellular ribosomes to direct synthesis of viral proteins. Viral proteins go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface. Viral surface proteins are made and processed in the cytoplasm and also travel to the cell surface, where they combine with the encapsulated nucleoproteins to form progeny viruses, which depart from the cell by budding. The virion now goes on to infect other cells. At present, vaccines to stimulate the production of antibodies against the virus and stimulate host cellular responses to ingest the virus remain the best strategy for controlling certain viral diseases, but they are of limited effectiveness against rapidly mutating viruses and are ineffective against viruses that cause the common cold and AIDS. Scientists at Xeris are working on new and better ways to prevent and manage viral infection by developing a platform of host cell-based targets to inhibit viral replication. To find these targets, we are using a patented technology called the gene trap. Xeris scientists transfect each cell in a colony of identical cells with a retrovirus, a small particle that causes a random insertion into the cell's DNA. The insertion randomly disrupts the function of a single gene in the cell into which it is inserted. Collectively, these cells create a unique library of cells, each having a different single gene disrupted. Importantly, cells that require the disrupted gene to survive will die and are not identified by the gene trap. Thus, there is a selection against genes that are essential for cell survival and likely toxic if targeted with drugs. Only the more promising drug targets are identified. Xeris scientists then infect every remaining cell with a virus that normally kills all cells, and identify those cells that survive the infection. Since all cells would normally be killed by the virus, the surviving cells must have survived due to a gene disruption of a human host gene product that the virus must use or hijack to replicate. These specifically identified genes and their RNA and proteins, when turned off, prevent a particular virus from replicating. The Xeris gene trap represents a cost-effective and efficient platform for the discovery of targets and drugs to block viral infection. The Xeris approach could provide the keys to preventing and treating infectious diseases and future pandemics, whether made by nature or mankind.